Class, hope you are good, okay? So let us check this interesting question. Stay tuned, don't forget to subscribe. So the diagram below represent what? Two stages of meiotic cell division, okay? So we are given diagram 1 and diagram 2, all right? So these are the different phases of meiotic cell division. Remember that we have what? Meiosis 1 and meiosis 2. Meiosis 1, we have prophase 1, metaphase 1, anaphase 1, telophase 1. Meiosis 2, prophase 2, metaphase 2, anaphase 2, telophase 2. You understand? So the examiners, they can take each and every phase belonging to meiosis 1 or meiosis 2 and they ask you questions. That is why we say be able to identify the phases. It is very much important for you to identify the phases and you must be able to know them. Yes, be able to spot that, oh, I see metaphase 2. Oh, I see anaphase 2. By this way, you will smash meiosis. And the way they ask questions, it is little bit tricky but easier. All right. So now let us go to the diagram. Now let's analyze the diagram first. Yes. So we are given diagram 1 and diagram 2. Okay. I see that. Diagram 1. It is the phase whereby there is what? The process there, whereby it will later on introduce what? The genetic variation. All right. So this phase, good people, what do we call it? Prophase 1. Why? Because everything there, good people, it now starts to prepare itself. You understand? We have what? We have crossing over there. You can see that letter A, it represents the collective process there of one chromosome and another chromosome, whereby they paired themselves in homologous pair. You understand? Whereby the two sister chromatids, they will overlap at a point called chiasmata, or other chiasma, to exchange the genetic the genetic material. You see, so now we can see that we have what? The formation of the spindle fiber. You understand? The nuclear membrane start to disappear and the chromosome, good people, they start to pair themselves in homologous pair. You understand? So now it tells me that diagram 1, it represents prophase 1. You see, this is prophase 1. So now let us check what? Diagram 2. So diagram 2, good people, we can see that the spindle fiber extends across the whole cell. And we can see that, okay, each and every chromosome there, good people, it has its partner. You understand? So one chromosome is dating with another chromosome. Hence, they are paired in homologous pair. They are the same in size. You understand? Same position of a centromere. Same position of a gene. You understand? Same size also. So you can see that this is what? Metaphase 2. There is a random arrangement of chromosome there. You understand? Yes. So you can see that diagram 2, it will be metaphase 1. You see, if it was metaphase 2, good people, don't confuse, because I know most of you guys, you confuse metaphase 1 and metaphase 2 with anaphase most of the time. So that is whereby we have a chromosome here, chromosome. So good people, metaphase 2, the chromosome there, they don't have the partners, they are single. So that is why we say chromosome, they pair them singly. You understand? So please don't confuse metaphase 2 and metaphase 1 differences. All right. Okay, now let us check what letter A. Okay, we are given that letter A, good people, it represents the process. So this process, good people, it is crossing. So we are given crossing over. Okay, we are given crossing over. Yes, and letter B, it represents what? The structure that gives rise to the formation of the spindle fiber, which is what? The centriole. You see. So C, it represents what? The spindle fiber. So this is the structure that join itself to the centromere. You understand? Yes. So this fiber. This will be the spindle fiber. You understand? So centromere is this, which is the structure that attaches the two chromatids together. So now D, it represents what? Homologous chromosomes. You see, these are homologous chromosomes. You understand? Please be able to identify the faces, good people. Yeah. So now we are given what? The question, they say name structure B. So we said B, it will be the centriole. So it will be the centriole, while C it will be the spindle fiber. You see, spindle fiber. Then you'll get a 1 mark, 1 mark. So please, I advise you good people to get these marks, 1 mark, 2 marks, you see, 3 marks, because at the end of the day, they will close the question whereby it will carry more marks, you understand? So if 1 mark is showing you flames, what about that one question? Please. Now, B, okay, B it is the central, yes. C, it is the what? The spindle fiber. Okay, now they say identify the face represented in diagram 1. You see, let's say you don't know the face. You will see Saturn. You see, so the face represented in diagram 1, good people, it will be what? Prophase 1. Let's say you are good at identify what? Identifying the faces. Alright, when I just say prophase, you are not sure what it is prophase 1 or prophase 2. Good people, prophase 2 crossing over does not take place. So it tells me that prophase 1, it is the one whereby crossing over is taking place. Please, 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 please. Alright, don't just say prophase, you'll get it wrong. So it will be prophase 1. Yeah. So now they say give 3 reasons for your answer to question 2.1.2. Okay, why are we saying this thing? It is prophase 1, good people. Okay, we identify that this one here, so that crossing over is taking place. You see, crossing over is taking place. Okay, and what else? What else? Spindle fibers begins to form. All right, if you can check here, check here. Check here. You see, check here. You see, so it tells me that the spindle fiber start to form. So here, spindle fiber start to begin, right? And what else? You see now, check here. The chromosome now, here, check. They pair themselves in homologous pair. You see, they align. So it tells me that chromosomes start to align themselves in homologous pair. Yes. And what else? It was 3 marks. Okay, and you can add that the nuclear membrane start to disappear. Yes, you can see here, this is the nuclear membrane here. You see, the nuclear membrane start to disappear. See, yeah, you can see start to disappear. Yes. Okay, now they say describe the process taking place at A. Good people, process taking place at A, so that this one is crossing over. So, and again, be able to describe the processes. Wherever you see prophase 1, good people, think that they will ask you why it is important. All right, describe it. Which phase? You understand? What is that point whereby the 2 chromatids overlap? Those are the questions that they like more when we are going to focus on what? Prophase 1. Yes. So now they say describe the process taking place at A. You know that here, the process here, good people, we are going to describe what? Crossing over. You see, we know that during prophase 1, good people of meiosis, that is whereby the homologous chromosome again overlaps at a point called chiasma to do what? To exchange the genetic material. To exchange the genetic material. Yes. So that is how you are going to get that remarks. Now they say identify the face represented in diagram 2. So the face, we said it will be what? Metaphase 1. Yes. Here we're going to have what? Metaphase 1. You understand? Now they say describe the differences in the event that they take place in the phase mentioned in A. So again, we set diagram to this metaphase 1. Yes. Now the question is saying describe the difference in the event that take place in the phase mentioned in A. A good people, we set it is metaphase 1 and the same phase during mitosis. Oh, this question, it is the same question whereby they say give the differences between metaphase 1 and metaphase 2. The same phase during mitosis, it will be what? Metaphase 2. So here we are going to give what? The difference in what? Metaphase 1 and metaphase 2. So we just say here in metaphase 1, the chromosomes in metaphase 1, the chromosome pair themselves in homologous pair. You understand? Then you say and the same phase during mitosis is that the chromosome pair themselves singly. You see, at the equator of the spindle fiber. Yes, you can end up by saying that. Yeah, so here we are just giving what the differences between metaphase 1 and metaphase 2, because the same phase during mitosis we are going to talk about what metaphase 2. You understand? So please, in metaphase what? The chromosome pair themselves in homologous pair, while the same phase during mitosis, the chromosome pair themselves singly. Yes, so that is whereby you are supposed to get that 2 mark. Right. Now let us check. Okay, let us check something. They say describe the results at the end of meiosis if chromosome at D fail to separate. Okay, let us check. Sorry about that. Okay, let us check. D. So good people, this is not non-disjunction. This is not non-disjunction. You are required to check what is going on there. You see, you are required to check what is going on there. Let me clean this. Okay. Yes. So you are required to check what is going on, good people. You can't say this is what? Non-disjunction. You understand? Actually, we are given what? We are given metaphase 1, and we saw that the chromosome at D, they fail to separate. You see? Let us check again. Check here. Okay? Let me show you. What is going on here? Okay. Jesus Christ. Okay. Yes. So check. Let's say. Okay. You see here how many chromosomes do we have? We have 3. There. And this side, how many chromosomes do we have here? It will be 5. 1, 2, 3, 4, 5. Yes, it is 5. So now they say describe the result at the end of meiotic division if chromosome D fail to separate. So let's say now this is metaphase 1. We go to anaphase 1. You see, so anaphase 1, we know that is whereby we are going to have what? The chromosome being pulled to the opposite poles. Whereby the spindle fiber shortened. So it tells me that this 3 chromosome will go away this side. And 5 chromosomes will go this side. Then when we go to telophase 1, that is whereby we are going to have what? A nuclear division, or the splitting of the cytoplasm, whereby cytokinesis is taking place. So it tells me that that is whereby we are going to have something like this. So it will be 2 cells two daughter cells that are being formed again. So it tells me that this cell, it will contain 3 chromosomes and this one will contain 5. So at the end, again, we know that those cells, they will not start to do what? They will not start to divide again. Again, they will not start to do what? To divide again. So it tells me that this one, again, with 3 chromosomes, it will not start to divide like this. The chromosome number, they will remain constant. It will be 3. It will be 3. And this one, it is 5. See. So plus this one is divide. Okay, sorry about that. It will divide. So it tells me that how many daughter cells am I having? 4. So that is whereby at the end, again, of meiotic division, again, I'm going to have what? 4 daughter cells. So that is why I'm going to say 4 daughter cells will be produced, or rather will be formed again, will be produced. And that is whereby you say 2 daughter cells will contain 3 chromosomes, and the other daughter cell will contain 5 chromosomes. You see, so this is what you were supposed to check. You just check, or okay, D failed to separate, which means one. You see this chromosomes you see here, this guy and this guy, they were together. They didn't separate. If it was just normal, we know that if it was normal, agree it is 1 2 3 4 1 2 3 4. It tells me that this one is supposed to go that side having what? 4 chromosomes, and this was supposed to go this side having what? 4 chromosomes. So there was an extra chromosome that was brought this side, you understand, causing the results at the end of the division, whereby we're going to have 2 daughter cells, you understand. We're going to have 4 daughter cells being formed, whereby 2 daughter cells will contain what? 3 chromosomes, and the other daughter cells will contain how many chromosomes? 1 2 3 4 5, you understand. So that was the question being asked. See, now they say describe the process of DNA replication. This one, good people, we have covered it when we are doing DNA. You can go and visit our previous videos. DNA replication, the DNA double helix unwind and unzips to form 2 separate strands. Both strands serve as a template, you understand, using free DNA nucleotides from the nucleoplasm. Each strand of DNA is now complementary. That is whereby adenine is paired with thymine, guanine pairs with cytosine. So this result to what? 2 identical DNA molecules being formed. So that is how you are going to grab that 6 marks. So good people, please don't forget to subscribe to our YouTube channel, and we are also available on what? TikTok. Please, Philip Junet Academic Support. We are with you, good people. All the best.